right, so I'm going to start off really talking about kind of the nuts and bolts fundamentals of the phosphoflow technique. And if you guys have any questions, or if you've already done this before and you kind of have a little bit of a more detailed question on something that I'm going through, feel free to jump in there. Either everybody's tired or everybody's just really intense this morning. So we'll see how it goes. But so again, I'm going to start off with kind of the real fundamentals, and then like Gary was saying, Sean's going to jump you right into the deep end. I was laughing about the lineup that we have going from here's like 2 color, you know, and then Sean will go 35. So it's good though, you'll get a full spectrum of kind of what we do here. So you know, the question is why phospho and why flow cytometry, and why jump out of your screen. So the technique itself is really all about looking at signaling inside single cells, as Gary was talking about. I just lost it. Let's go back in. So why look at signaling? So signaling is really kind of at the pinnacle of what happens inside your cells. So if you kind of look at the different cell events that you might have over time, maybe for a long-term type differentiation event, that might take days or weeks depending on what cell subset or cell type you're looking at. Events like protein translation or secretion might take hours or days. And then you might have events like transcription, again, on that same kind of time scale. And then phosphorylation, really, though, is that thing that happens immediately as soon as something binds to the outside or potentially to the inside of the cell. And so you might have a cytokine binding to the outside of the cell that we'll be talking about a lot, inducing JAK-STAT signaling and leading to transcription inside the nucleus. And so we really see phosphorylation as kind of that immediate first readout that you can get from a cell as to how it can respond to its environment. And also the quickest thing that you can measure, so instead of having to wait for a long-term kind of readout, you can get an immediate response and an immediate readout. So flow versus kind of the traditional methods looking at biochemistry, normally you kind of, as Gary alluded to, you look at populations. Again, in flow, you get information for every single cell going through there. Normally, the traditional methods, too, are single parameter methods, whereas flow, obviously, you can do many parameters per cell. In flow, again, you can look at heterogeneous primary cell populations, and you can analyze really rare cell subsets. So again, people that have done this before can appreciate, that you can look at very rare cell subsets like dendritic cells or something that might only be, let's say, 1% of the total population in your sample. And on the scale of things, it's actually quite rapid and scalable, too, to hundreds or maybe even thousands of samples, whereas you really don't want to be doing that many when you're doing a Western blot. So again, the traditional method, you take a bunch of cells that you might have, you aggregate those up, and you put them all, and you mash them up, and you get one value for that entire population. The beauty of doing it with flow is that now you can take those same cells, you run them through the cytometer, and now you get a value for every single cell. So you might have a population that's quite homogeneous, or you might start seeing that you actually have some outliers in your population. You might have, even in what you think is a homogeneous cell line, you might have a few events that are a little bit higher, a little bit lower. You might have a subset that is responding differently. By being able to measure a lot of parameters per cell, you can start to throw a lot of different antibodies on there. So now we're getting information for every single cell, and you can have, let's say, 10 different fluorophores or 10 different mass labels on your cells, and you get a measurement for each of those now as it's going through. And so what that really enables is what I was mentioning, kind of this ability to look at the heterogeneous populations of cells. So now you can do a combination where you're looking at different surface markers, let's say, and also different phosphoproteins. So now for every single cell subset in there, you can measure, CD3, CD4 to identify your T cells, and then you can look at the phosphorylation of, let's say, STAT1 or STAT5 within those subsets. And you can also have, CD19, CD20 in there to identify your B cells. You can identify your monocytes. And so again, from each tube now that you're running, you're getting this large multiplexing of the data where you're measuring all these different subsets, and you're getting all these different signaling information out of it at the same time. So just as a kind of a graphical example, if you had 3 different cell subsets here, you can have 3 antibodies to identify each of those subsets. And then within each of those, you can look at, let's say, 3 different phosphoproteins, and you can measure out those different levels. So you can start to imagine that, if you have 5 surface markers and 5 phosphoproteins, now all of a sudden you're getting, let's say, 25 different data points from each single tube that you're running through. And then obviously if you add different conditions to those cells, let's say another 5 conditions, you have 5 times 5 times 5. So all of a sudden, just in 5 simple tubes, you're actually getting 125 data points about the particular cell sample that you're looking at. And this is that huge kind of multiplex advantage that we really see coming out of this, that you can multiplex at the level of doing different stimulations and different conditions, but then also at the staining and the revealing step where you can do different cell subsets and then also different signaling molecules at the same time. And this is really where the power comes in. And I'm always surprised to see a lot of places that actually work up the phosphoflow technique. They get it working really well, but then they just decide that they're only going to look at CD4 positive T cells in response to STAT5. And they always look at our stuff and are like, oh, how do you do all this? I say, just add CD20 in there and you can look at your B cells at the same time. But they really like to optimize in on one thing, and I think the real advantage of this is, each one that you add on there is, per tube, now you're getting an exponential multiplication of your data. So you add 2 surface, 3 surface, 4, all of a sudden you're getting that many more data points out of each tube. So again, in the ability to look at those rare cell subsets, I think is a real key advantage, obviously, of flow. So being able to look at, those couple yellow cells that you might have in your peripheral blood or in your mouse splenocytes, you can get that 1 or 2 cells out of that entire heterogeneous bunch and really zero in on those guys. So the overall technique, just to give you kind of a preview of how this works, we're going to put it in the context of cytokine signaling, since that's what we're going to do, at least partially in the experimental work and what we've done a lot with, since it is kind of the fundamental signaling that happens between immune cells. So looking at cytokine signaling, the cytokine signaling pathway is relatively simple on the scale of things, where you have the cytokine that binds to the outside of the cell, leads to dimerization of its cytokine receptor, which activates the JAK kinases that are docked on the inside of the cytokine receptors. And then you get activation of the STAT transcription factors, and these are basically the downstream effectors. Those go into the nucleus and cause transcription of your target genes. And so overall, it's a pretty simple system. There's only a couple parts to it. There's the receptor, the JAK, and the STAT. And so even with that kind of simpleness of it, there's actually a ton of complexity due to expression of the different receptors on different cell subsets, the abundance of different JAK kinases and STATs also within the different cell subsets, and obviously localization of the cytokine. Cytokines don't just get ejected into the blood and float around everywhere at a high concentration. There's always some kind of localization. If you have some kind of immune event going on, that's not just going everywhere. It tends to be localized. So even though the system is relatively kind of simple from the signaling standpoint, the multiplication that you get from all the cell subsets and all the different abundances of these molecules really make for a complex system. And you can't just get these really simple maps of IL-2, leading to JAK3, STAT5 that you see in all the reviews out there. Just don't really hold when you start to look at all the different cell subsets and all the different responses that you actually get in these different cell subsets. So then at this point is where the kind of the phosphoflow technique takes over. And so the first thing that we do in the technique, and we'll go into kind of how we got to this point, is fix the cells with formaldehyde. So the point of fixing the cells is basically to stop everything that's happening inside those cells. So you had signaling going on. Signaling is obviously a pretty transient event. It doesn't just go on and stay on. It's something that will go up and it'll go back down again as you start to relax the cell away from that stimulus. So the first thing that we have to do is kind of freeze the cell and stop it from doing anything else, and that's what we do with formaldehyde. The next thing that we have to do then is to open up the cell for our antibodies to get into the cell. So you'll see we tried different permeabilization reagents, and methanol turned out to be kind of our favorite and seems to work the best generically. That being said, there are different perm reagents, and you can play around with different reagents to try to find one that works best for your particular system. Then you come in with your different fluorescently labeled or mass-labeled antibodies to identify now the phosphorylation events inside your cell, and you run that on the cytometer. So typically what you'll see when you run some of these samples is that in an unstimulated or untreated state, you'll typically see a little bit of antibody binding, just like you would with any kind of surface marker, depending on your titration. You might have a little bit of background, and so if you just have autofluorescence in the dotted line, green is your actual sample. You might have a little bit of signal, and this is pretty typical. But then when you induce your sample and you have a lot of phosphorylation, now you have a lot of specific binding inside the cell, you should see a large increase now, obviously, in that binding. And so the phosphorylation basically correlates to the level of fluorescence that you have in your sample at this point, so it's really easy kind of to look at. An increase in fluorescence means you have more phosphorylation inside your cell for that particular phosphoepitope. So the phosphoflow and Western do correlate nicely. So if you actually look at some of our early work, just looking at STAT1, STAT5, and STAT6, looking at a few different stimulus conditions in our good old U937 cells, sadly, we're actually not going to run our U937s today. So this is, you guys are getting kind of the 2.0 version of the technique because we wanted to do a little bit on the CyTOF. Typically, we start with this cell line, and I really highly recommend anyone that wants to try this out to use these U937s. I still haven't seen an experiment that was run sort of somewhat correctly not show a good response to this. So if you want to start somewhere, this is the place to start, just to get, really dial in the technique and make sure that it works. So to look at the comparison to normal Western blotting, if you see a band in a Western, that basically correlates to a shift to the right in your phosphoflow. So you can see with interferon gamma, you induce STAT1, and you see a band, and you see a shift to the right. With STAT5, you can see you actually get intermediate levels and then a high level. And I'll show some more where it's actually quantitative, a little bit more detail. And so what you'll hear a lot that we describe kind of this quantitativeness is with a very simple metric, fold change, where it's just the fluorescence after stimulation divided by the fluorescence before stimulation. And so this is just a way, this is not necessarily to say that the phosphorylation of that protein actually increased 5-fold. That's to say that the fluorescence that we're measuring or the mass label that we're measuring went up 5-fold. And so it's something that some people always try to eke out, is that truly the amount that phosphorylation is going up? It might be, but it's something that we've never really validated, and it's not that trivial to validate, actually, that you're getting kind of a one-to-one binding perfectly for each of those, and that you're at full saturation and all these kinds of things. So the fold change is really a metric that we use for phosphoflow as a way to just compare between samples. It's going up 5-fold, it's going up 3-fold, 2-fold, 10-fold, whatever it might be. So with this, we have a nice quantitative single-cell analysis tool now to look at intracellular signaling. And again, this lets you now look at those heterogeneous cell types that are really what we were always interested in, is looking at a heterogeneous sample of peripheral blood or cancer sample in the peripheral blood where you might have different cell subsets that you really want to look at. What's the signaling inside those? So how did we get to this protocol? So what we originally did, it's a long time ago now in cytometry, was to look at basically different fix and perm conditions to see which one would give us the optimal signal to noise or the optimal fold change in this case for a few different phosphoproteins. And this wasn't exhaustive. This was basically some of the first phosphospecific antibodies that were validated for flow or were being validated for flow that we tried out back then. And now there's, probably on the order of a good 40, 50, maybe even more that are actually validated and are conjugated already and available from either BD or CST and some other vendors too. So basically what we did in this was try 10 different conditions. The first 3 we did no formaldehyde fix, and then the rest we did a fix on those cells. The first 3 were organic solvents, basically, where people that have done, let's say, DNA analysis and things are probably familiar. You can just do straight ethanol as a fix and a perm, basically simultaneous. That kills, obviously, the cells when you dehydrate them completely. They're not going to survive in no water. So you can actually just use these directly as kind of a fix and perm, basically, at the same time. You can also do a fix before and then add those. And then we also did detergents, Triton, and saponin as well. And what you'll see right off the bat is mostly that these red-colored samples are the ones that are showing the largest fold changes. And again, these we just induced with their cognate stimuli. And what you can see in general is that number 4, which is the formaldehyde fix followed by the methanol perm, kind of stood out as the one that was generically kind of the best. But you'll notice also that even some of the ones where we did no fix in the blue turn out quite well, and also that between methanol, ethanol, and acetone, there's not necessarily a massive difference for some of these either. And also that you can use saponin, and some of these results actually have gotten better as the saponin has gotten more purified by some of the vendors, where you can actually get quite good results too with that. So depending on what phosphoprotein you're interested in, there is some optimization that you can do. And so this is something we've done recently really to dial in a particular phosphoprotein as well. Really just, messing with the different conditions can actually give you a lot larger fold change. So, if there's one thing that you really, really, really want to look at, don't just take this protocol and just assume that that's the best thing there is. So really mess around with, different fix, maybe higher or lower, different perm reagents. It's definitely worth your time to do that because you can change something that might be a really small, two-fold change into a 3-4-fold change or something like that. It'll make you feel a lot better about your data. It'll make your statistics 1,000 times better, and you'll just by eye, you can look at that and know that it's actually a good shift. So, again, even though this is a kind of a generic protocol, I really urge you, if you are doing one specific one, to really optimize in for that one specific one. So again, the formaldehyde followed by methanol is what I would call kind of the most general, and is typically what we use because it is the most general. And again, you can get a little bit better with some, but some of those also have trade-offs where when you use that other technique, for instance, if you use something like a saponin, you might be able to measure some of the more membrane proximal events sometimes a little bit better, but then all of a sudden you can't measure the stats at all. So you won't get any response at all in there when you use detergents, where the alcohols or some kind of harsher kind of denaturation is required, and you really have to use some kind of methanol or ethanol to reveal those stat transcription factor phosphorylation events. So there are trade-offs on those, and you have to be aware of those obviously too. So again, that's why we typically use the methanol, because we're interested in cytokine signaling. It's kind of the bread and butter, and that we would rather lose a little bit maybe on some of the other things that aren't quite as important as looking at the stats, let's say. So the kind of optimization of some of these steps, and you can start to see a little bit of what I was talking about. If you first look at the fixed time, so this is how long are we sitting in there with formaldehyde. We just did 1, 10, and 30 minutes. You can see there's not a huge change, but there definitely is a subtle change. So you can see with the short fix going into longer, you can see a tightening and an increase of that positive peak. So again, these are unstimulated in the white histogram, and then the stimulated samples in the gray. And so it's subtle, but there's a pretty significant increase in that signal. And so that's something, again, that if you want to only look at P38, maybe you want to fix for a little bit longer time. The converse happens, though, on the stat1, and I don't have a line drawn on here, but you can take my word for it. It actually is going down as you fix for a longer time period. So again, this is basically the moral of the story on phosphoflow, is that if you're increasing one phosphoprotein measurement, you're probably decreasing some other ones. And so that's just the way that it goes. We've done a lot in this lab, and I would say that's pretty much always the trend, is that you'll dial in and you'll be like, this is great, I can get 20-fold on P38, and then all of a sudden your stat1's like 2-fold. So it's always kind of a give and take on that. And again, what we've done is kind of dial in that 10-minute time point for the fix is the one that, again, is most general, but isn't the one that is always the best for everything. The fixed temperature, room temp versus 37, we haven't ever really seen a huge difference with this. These are basically identical to each other. So typically what we do is fix our cells. We'll take them out of an incubator. We'll throw the fixative on, and we'll just leave them there on the bench top. And at that point, there's really not much need to kind of go back into the incubator. The methanol perm time. So one of the big advantages of methanol relative to some of the other perm reagents that you might use is that you can actually store your samples in methanol for a really long period of time. And here we actually just did some storage at -20, so this wasn't actually at -80, this was only at -20. And you can see even for a week or 5 weeks, you still have pretty good signal there, and you're actually getting a pretty good fold change. If you store these at -80 actually in methanol, you'll see this hardly change actually. And so this is something that we've done quite a bit, where you can store your samples in methanol for months and months, and basically aggregate up samples and then do all the staining on the same day and all the analyzing on the same day. And that's obviously a huge advantage, because you don't throw that error in now of every single day. Let's say I get one clinical sample, I stain that, I run that. The next day I stain, I run, I stain, I run. You're going to have a lot more variability just from kind of day-to-day wobble on things like that, as opposed to I do my stim, I fix perm, throw it in the freezer, fix perm, throw it in the freezer, and then let's say 3 weeks later I have 20 samples, and now I stain them all and run them all on the same day. You're going to feel a lot better that they all got the exact same surface cocktail. the same phospho cocktail and got run with the exact same instrument setup, settings, laser flux, you know, whatever, depending how much control you have over your cytometer, that makes a big difference. So again, that's a really big advantage that you can't do in something like saponin. And there are other ways around it where you can freeze your samples, maybe post-fix and things like that. And you can feel free to ask some questions later of people too to see if they have any tips on things like that. And those are things that people typically kind of just optimize on a per user kind of basis, depending on what their workflow is and how they get their samples and things. You know, where can I throw this in the freezer? And there are different times that you can throw it in the freezer too. And then for the staining time, basically we looked at 15 minutes to 1 hour. And what you'll see in general is that you'll get an increase in the positives, but you'll also typically see kind of an increase in your background staining as well. And so just by the nature of it staining a lot of intracellular epitopes, you'll notice that as you do longer stains, you'll typically have a little more increase in background than you're used to maybe on just a live cell kind of doing a surface stain. On a live cell surface stain, you can basically throw in as much antibody as you want. The stain's going to be clean. You really don't even have to wash your cells half the time if you don't have a crazy titer or the recommended titer that they tell you to use. But with phosphoantibodies and when you're staining with surface antibodies now, once the cell is permed, that's a very different story. So especially when you're starting to use things like some of the tandems and stuff too, your PE-Cy7, your Percy PE-Cy55, some of the more red dyes too, like Alexa 700 and things like that. Once the cell is permed, those antibodies have a much higher propensity to just stick to the stuff inside the cell. So if you throw in your normal like 20 microliters of that surface marker, you're going to get a signal up at 10 to the 3 and just everything is stained and you won't be able to tell anything apart. So it's really important to kind of be aware of your, once you're dealing with the perm cell, that it's a very different kind of background than it is on a live cell where it's only seeing the stuff on the surface. So speaking of the fluorophores, we just did originally a little comparison, looking at, again, what was kind of the standard at that point, the two Lexodides 488 and 647. And these are still the two that we typically use for the most part for almost all of our stains. There are also now, you can get the PE, Percy P, PE-Cy7, Pac Blue, all these guys now on the phosphoantibodies, so you can definitely mix and match. But just to kind of compare on these, you can see in general, if anything, the Alexa 647 most of the time will be the one that's better. And then sometimes you'll have ones where, between the two fluorophores, there's not a big difference. And so, again, you have to titrate your antibodies to get, kind of the optimal signal to noise. But in some antibodies, you're just a little more, dependent on having that red dye to get a little more signal out of it. Some of them aren't as dependent, so you can, again, tailor your cocktails to match that a little bit to ones where you're not losing as much, let's say going on the 488, and then the ones where you are losing signal, make sure to use that 647. So again, the technique is really quantitative. So here's looking at a dose response of interferon gamma inducing STAT1, again in the U937s. You can see the Western blot getting in the induction. Then you can see by phosphoflow here with the 647 and the 488. You can see a really nice induction. You plot those both out as a percent of max, and you're basically getting those to overlap directly right on top of each other, which is what you would hope and what you want to see. So again, the technique is very quantitative, and it's not just kind of a qualitative, does it have phosphorylation or not? No, you can actually measure those intermediate amounts. You can do dose responses of drugs. You can do dose responses of cytokines. You can do, these kind of quantitative techniques. And we actually use it quite a bit for drug discovery and drug screening now, too. And so here's just a sample plot of people that have done enough flow. We'll have seen plenty of these, but people that haven't, it's good to see kind of what a dot plot looks like. So in this case, we've overlaid 3 samples, and we did 2 phosphostains, STAT1 and STAT6. And you can see your unstimulated sample here in the bottom quadrant. And then you can see your interferon gamma induces STAT1, and your IL-4 induces STAT6. So this is really nice. You can see that the two stimulation conditions are basically orthogonal, where one induces STAT1 and one induces STAT6, and they don't really show up in this double positive zone because they're only inducing one pathway at a time. So again, you can imagine, if you do 4 of these phosphos at a time, or you do a bunch of cell subsets, you can just get a lot of information now, and you can get a lot of subtle information too. Again, they aren't inducing that other protein at all. They are inducing maybe it a little bit or something. So you will see stimulations that do a little bit of both, depending on which phosphoproteins you're looking at. And that gives you information that lets you separate out those two signaling, those two cytokines, let's say, where one might induce STAT1, one might induce STAT3, and a little bit of STAT5 or something like that. And so you can get these subtle differences that help you identify the characterization of that different cytokine. So moving kind of from the initial cell line work to primary cells, which is where a lot of people are interested, is you need a few different things. Number one, you need your surface marker antibodies. And normally we actually had a whole talk about doing surface markers and kind of optimizing on those, but we can give you the reference for the paper to look at for that information. And then you have to know a little bit about your stimulus. And I'll just kind of jump through a few of these things just so you can get some idea about cell type specificity, pathway specificity, the dose, and the time point. So again, it's a very complex system, and all the things that you see, in cell line literature don't necessarily always hold when you start to go into the primary cells. And that's one thing that I always try to caution people, because they say, my, NK cell line has a great response to this thing, and then I go into my primary cells and it doesn't have the response. They're like, what's wrong? And I say it's because they just don't respond to that in primary cells. We've done that enough times. So there are a lot of things where, your cell lines are cell lines for a reason. They grow forever. They're cancers. They have a lot of aberrant signaling pathways. And so, a lot of time that happens where people think that the phosphoflow technique is flawed or something, and it's like, no, that's just not there in your primary cells. And so that's something to always really be aware of. So what we did in this case was actually just look at signaling in some mouse cells. This was some early work that we were doing where we took out splenocytes, homogenized them, and did signaling, where we actually compared it to their response if we just injected the cytokine directly into the mouse, then removed the spleen and fixed the cells really quickly, and did phosphoflow. So the first thing, again, is you need to define your cell subsets. So if you take a kind of a typical scattergate of some splenocytes here, and you look at B220, which is a B cell marker, and TCR, which is a T cell marker, you can see your B cells and your T cells here identified. If you take those double negative cells, you can go into C11b, which is monocyte, kind of macrophage, granulocyte marker, and identify neutrophils, which are the C11b highs, and then most of the macrophages, DC, some other cell subsets that are in this intermediate population. And so right off the get-go here, using three different surface markers, we actually identified 4 different cell subsets in that case. So you might obviously have some surface markers that resolve multiple populations, and so that's always kind of a bonus. If you have something that's got a high and an intermediate and a low or something like that, now you put in 1 surface marker and you get 3 cell subsets out of that. So if you have a few of those, all of a sudden now with even a 3-color stain, you might be getting 6, 7 populations or something out of that. So this was originally kind of a lot of this work. When I look back at this, this was pure brute force. You don't even know how long it would take to actually generate some of these figures that we did, where basically these histogram overlays, we would generate these in FlowJo. Then we would literally hand color in the fold change using Illustrator. So we'd actually make a palette out of a heat map program, basically where the fold change would correlate to some color, and then we would go in there and drop that color basically on all the histograms. And you'll see today when you're using Cytobank, that'll take you probably about 30 seconds to make that thing, and that literally took us, would take us a week or something to make that. And so it's a big change now going through. Now we analyze 96 well plates for 6 surface markers in literally like 20 minutes or something like that. Generate out a heat map, generate out the histogram overlay, and we're done. And so the times have definitely changed since we were first doing this. So what you can see, though, in this, just to kind of focus on a couple, it's not really that important, is just these concepts of you might have something like interferon gamma, again, that's specific to STAT1. It doesn't induce STAT3, 5, or P38. So again, it's pathway specific. It's only doing one thing inside those cells. You might have other stims like GM-CSF that are more cell type specific. So again, this is going to induce these macrophage and neutrophil populations that actually have those receptors, where you'll see an induction of GM-CSF, but you won't see it in the B cells or the T cells. So again, it's not, one stim doesn't do everything to every single cell subset. They have selectivity for particular cell subsets. And then picking, kind of looking at some of the dose responses of some of these cytokines in the primary cells. Again, you can look at the in vitro response, IL-10 induction here going from a low concentration to a high one. You can see that you have a good response in B cells and the CD11B intermediate population, but you don't have response in the highs in the T cells. So again, cell type specific. the nice thing is that actually even if you do this in vivo and you inject these different amounts of IL-10 into the mouse, again, you directly fix the cells out of the spleen, you can actually see a very similar EC50 if you kind of average out about 2 mils of blood in the mouse, that the values are quite close. And for some of these cytokines like IL-10 that are very specific to immune cells, this is typically the case, and we did this with quite a few. But what's good about that is that you see the same cell subsets respond in vitro and in vivo, and you see it relatively the same EC50. So you feel good that what you're doing actually in those cases ex vivo in those mouse models is quite good and correlates quite well to the in vivo work. But again, you want to be picking a dose, obviously, too, that is enough to induce that pathway all the way. So those are things that you need to kind of measure empirically, figure out what's the dose that I need of this. Typically, you can start at a really high one, obviously, and then dial back, and see how much do I really need. So then you're not breaking the bank if you're throwing in, microgram per mil or something of a cytokine. You don't want to be doing that on a lot of samples. The time point is kind of an interesting one, too, and is I think, sort of underestimated in value a lot of the time. With signaling, again, it's transient. You have to pick the right time point to look at. And one time point doesn't necessarily fit every single thing that you might be measuring. So in this case, I'm just going to kind of zoom in and look at the histograms a little easier to see, where, again, we did this little comparison in vivo and in vitro. Number one, you see actually some different cell subset response in vivo versus in vitro. But you also see a different time kinetic on that as well, where in vivo we actually get a super rapid response that decays already by 15 minutes, whereas in vitro you actually see that come up at 15 and start decaying at 30 and gone by 60. So, if you were doing this experiment and you said, your typical time point was 5 minutes, you wouldn't see anything. And there might be other pathways, too, that might be up at 5 minutes and gone by 15. So if you just take one generic time point of, say, 15 minutes, you might be missing a lot of things. And so, again, it's really important to look in there and to look at, 15 minutes or something is a pretty good spot to start. Normally, you can get the things that are really quick. You can kind of get the tail end, or things that are a little bit slower, you can see the front end. So it's a good place to start, but it's definitely not kind of the one-size-fits-all time point. And then one of the nice things, too, with this, again, is the nuance, I think, looking at some of the different cell subsets and how they respond to these cytokines. And so this is an example, again, from some mouse work where we're looking at the Treg subset in your CD4 positive T cells. And so if you actually looked at TCR, CD4, gated on those guys, and you had your CD4 positives, and now you look at CD25 on the y-axis, which is the high-affinity IL-2 receptor, and then STAT5 on the x-axis. What you can see is you start to dose in IL-2 from low to higher doses here going across. You can start to see this population right here of the CD25 highs are the first ones to basically respond to IL-2, which is what you would expect, obviously, if you stain for IL-3 receptor or IL-7 receptor or something like that. You would hope that those cells that have the highest amount would be the ones that respond first kind of to that particular cytokine. And so you can see that nice induction, and then only when you start to get it about 100-fold higher do you start to see some of these more negative cells, which are the low affinity receptor, actually start to respond, and that's what you would expect is they have about 100-fold difference in their EC50 values for those different receptors. And again, you can actually see that same sort of thing too on the in vivo work. What was interesting in vivo was that we actually saw, again, this induction, but we saw a little bit of split at some of these doses too, where you actually only see a part of the cells respond, probably due to their localization in the spleen and their exposure to the actual injected cytokine. But overall, again, this is really kind of the fine nuance that you can get out of using this phosphoflow technique, using surface markers in combination with the phosphoproteins to really measure and get at those really fine subsets that you wouldn't see if you just took that whole bulk. You would see a little bit moving out. Once you add that extra surface marker and reveal what cells those are, it gives you a lot cleaner of a response and really starts to let you dial in to those different phenotypes and those different subsets. So the rest of the stuff that I have in here, and since you guys have the slides printed out, you can look at those there for kind of some of the references that we have and some of the things that you can look up. These, again, were some of the kind of early papers that are still very useful to look at for giving yourself some ideas of things that you can do to optimize and mess with. Definitely take a look, too, for people that want to do more surface analysis. Take a look at this paper that we had in JI where we did that in mouse samples, but the same holds true for human samples, and all the techniques that we have in there are very useful basically for optimizing out some of the surface markers, some of the things that you can see go wrong, some of the things that go right, just to give you a sense of that. Some surface marker antibodies are really sensitive even to once the cells are fixed. Other ones, are very affected by the perm reagents. So you really want to look at that. And again, since we're not going into that, I mean, the surface markers are by far the bigger problem than the actual phosphoproteins. So people that go into primary cells, 9 times out of 10, when I talk to somebody, they tried to do the same surface stain that they always do on their subsets. Oh yeah, I threw in my 6 surface marker antibodies. What titer did you use? Oh, what I use on live cells, it always looks great. They throw it in there and they basically just have these blobs of stuff that are just positive, off the charts positive in some of the dimensions. And the problem is because, yeah, they haven't titrated it, they haven't optimized it at all for the fixed and perm cells. So you really got to go in there and test those surface markers on fixed and perm cells. Don't just do your 10 stims, throw in all your surface markers, and just hope those guys are going to work, because they won't. I guarantee you they won't. So you're just going to be throwing away, a couple $100 of antibodies or whatever in that first experiment. So really, do the simple experiment where you take your live cells, you stain those, you take your fixed perm cells or just your fixed cells, depending on how you want to do it, and basically do the head-to-head comparison. Do a few different titers of antibody. Look at the live cells, how you resolve it, how's your gating look. Look at the fixed perm cells. Do I see those same cell subsets? Do I see the same percentages? If all of a sudden you only got 5% instead of 30%, something's not right, and so you can't just assume, oh, because there's a few CD4 cells out here, it's all perfect. You want to make sure that you actually have the right abundance of what you're expecting in each of those. So it's really important to do that comparison of kind of the live versus the fixed perm, and to feel good about that, that you actually have these surface markers staining the right thing, so that you don't go and do 6 months of work and you're saying these are my B cells when really you're just staining background or something like that. So again, these are just some tips that you can kind of take home. Again, checking out your stimulus, your cells, your cell subset. Again, don't assume all surface markers work, and don't assume all cells are represented equally before and after. And if you don't get a response, don't always assume that it's because something is wrong with the technique or whatever. It might just be because that subset doesn't respond in the primary cells. And then we also have some things in here for just looking at some different phosphoantibodies. Again, now there's a lot that are already commercially available, so it's a lot easier. But if you want to try to do a new one, here's some kind of tips too. And especially if you're trying to do some phosphos, you typically have to do a few different ones. And even with surface markers, you might have to do a few different ones. So with that, I'll stop, and I want to thank Matt Hale, who did did the in vivo work with me way back in the day, and he'll still be here today as well, and talk to you guys about some stuff too. So, and then we've had a close relationship with BD a lot too, in this early development, really, to get these antibodies working, and that was always a great collaboration. So, thank you. Yeah. Yeah, you have to be, so yeah, the question is how fast you have to be to process like the spleen or something in an in vivo stim. We got pretty good at that, actually, where we typically had 2 people going, so one person was lopping the spleen out and the other person would do the fixing. We got it going in about 2 minutes or something like that, so we got pretty fast, but that really depends more on the pathway that you're looking at, I think, and the kinetics of it. If you have something that it peaks at 30 minutes or something, it's probably not as critical that you get really fast at it. When we're trying to do some of these things that were up and down by 5 minutes, obviously that was a lot more critical. So with cytokines, you have a little more flexibility. So what we actually normally do is we would put the fixative directly in PBS and just actually do our homogenization of the spleen directly in the fix right there. So it actually was just you take the spleen out and you fix it directly by homogenizing right into that. So it worked pretty well. Yeah. Yeah, I mean, the in vivo, like I say, normally it actually, that's what we were surprised by, is actually when you look at that, the blood flow through the spleen, for instance, is extremely fast. I mean, you basically will probably pump through that spleen multiple times in each minute almost, So the actual circulation and distribution of the cytokine, we think is pretty much almost instant, actually, when we do those systemic IV injections. No, we didn't do the bone marrow, and so that one is just technically would be obviously more challenging. You know, how quickly you can get that out is the bigger hurdle there. You know, I think in that case I would probably just flush the marrow, you know, directly with the fixative basically, and just flush it out as opposed to flushing it and then, you know, spinning it and doing that kind of thing.
And for putting the cytokinin, would you think intrahumoral injection would be better, or would still an IV be?
My guess would be IV would still probably be almost faster actually to get through there, yeah, as opposed to kind of dissipating through the muscle a little bit more, yeah. Excellent. All right, so I think we're going to jump in the deep end now with